Hi all. In the previous two videos, we learned about transcription, RNA production, and translation, protein production. These are the basic processes necessary for gene expression. The production of correct protein is critical for maintaining health. Too much production of proto-oncogene protein can contribute to cancer. Production of incorrect tumor suppressor protein can also lead to cancer. Obviously, having the right recipe is important for producing the correct protein. Having a recipe call for wrapping a baking potato in foil is reasonable. A typo that changes the instructions to wrapping a baking potato in soil, well, that would be a bad thing. Mutations are typos, errors in the nucleotide sequence of DNA. Since they are changes to the nucleotide sequence of DNA, they are permanent and will be passed down through DNA replication in each S phase of the cell cycle. Changes to RNAs or proteins without changes to DNA are just temporary, but any change to DNA will be a change to the permanent record of that cell and all of its descendants. Mutations, changes to the nucleotide sequence of DNA. There are several ways that nucleotide sequence can change and several effects that those changes can have. Let's take a look. A base substitution mutation occurs when a single nucleotide is different than the original recipe. If that change escapes DNA repair and the complementary nucleotide changes to form a complementary base pair, then the base substitution mutation is made permanent. An insertion mutation occurs when one or more excess nucleotides are added to the nucleotide sequence. By contrast, a deletion mutation occurs when one or more nucleotides are omitted from a nucleotide sequence. Base substitutions, insertions, and deletions are all descriptions of the nucleotide changes that can be classified as mutations. Those nucleotide sequence changes may affect the protein's amino acid sequence. A missense mutation occurs when a mutation changes one amino acid in the protein. Sickle cell disease is caused by a missense mutation that causes a change of one amino acid out of 570 plus amino acids in the hemoglobin molecule. That one amino acid change causes large-scale changes to the hemoglobin molecule that can lead to whole body symptoms in the person. If a mutation causes the production of a new stop codon where there shouldn't be one, we call that a nonsense mutation. Many base substitution mutations result in neutral mutations. Neutral mutations are changes to the nucleotide sequence of DNA that do not result in change in protein production. While neutral mutations are inconsequential in the short run, in the long run they may have effect if another mutation happens close by that makes this first neutral mutation more relevant. One other type of mutation occurs from insertions or deletions in numbers other than multiples of 3. This leads to what is called a frameshift mutation. When the first AUG is found in the mRNA, that sets the reading frame, the sets of nucleotides that make up the codons that are read in translation. If an insertion or deletion mutation occurs, then the combinations of nucleotides are offset and every codon downstream of the insertion or deletion is thrown off. Since the reading frame is shifted, we call this a frameshift mutation. Frameshifts usually have major effects on the functioning of the protein because every downstream codon is affected. Neutral mutations are usually not very harmful. Other mutations, simple base substitutions, missense, and nonsense mutations, might have small effects, or they might have major effects on the protein and on the individual. Whether mutations have large or small or non-existent effects on the protein, they're still mutations, changes to the nucleotide sequence. In the last video for this chapter, we'll look at other prokaryotic and eukaryotic factors that can have impact on how chromosomes are replicated and how they're expressed.